So if it's super affinity, super affine, but still we don't really understand why this is. So, and I think there's a lot of cooperativity ongoing in that binding. Yeah.
How would you calculate if you have 1 millimolar and 10 millimolar if you attach the two? What would be the method to calculate the conjugated ligand efficacy? Since you mentioned delta G, so we should think in the word of delta G. So it's kind of not averaging or adding, or we have to go to another scale and calculate the delta G and then calculate the conjugated, but it still won't resemble the entropy what you mentioned, since the two parts will be together, so we gain one entropy term, and if you could calculate that, that's the non-cooperative. So every addition to top of that would be the cooperativity, which is some kind of a steric arrangement that's provided by the combined structure.
So the question was, how would you calculate this? It's probably the best question to ask, and you're the expert to answer it. I have no idea.
Yeah. My question is, you mentioned SCR matrices and analysis. What values are you put into these tables?
So you would look at IC50s or EC50s?
Mm-hmm. Yeah, that's interesting, since delta G is, you all know physics. Yeah. So if you would like to use the IC50, so it's better to have the PIC50 values in the table. Yeah. That's one thing. Then it would correlate with delta G that you are explicitly optimizing. Yeah. It's quite rare to see SAR tables in P activity values. Very rare. If you check one of these tables, you will find that there are not that big difference in between the numbers. Since going from PIC50 6 to 7 is equivalent in terms of delta G, then going from 7 to 8. I hope that you can still follow. So it's the same drop in the activity when you are moving from 1 micromolar to 100, then from 100 nanomolar, then from 100 nanomolar to 10 nanomolar. And these activity cliffs, so yeah, the motion of the thermal motion is about 5 kcal, 20 kilojoule per mole. So one unit in the P scale is 1.4 kcal, which is quite tiny. And then this is what you mentioned, activity cliff. And I was thinking that, okay, in general we are measuring in 1 micromolar. So if the activity cliff could be originated from 3 kcal per mole, which is in chemistry, as far as I understood, it's not a big deal at all. So if we are turning our tables or SAR activity tables into P activity values, and if we could measure on a longer scale, then we might have the chance to understand these activity cliffs. So if I had the values in P activity from 9 to 5, then I might be able to have a continuous scale and have better understanding.
I mean, this is then after the fact. So you discover the cliff and you describe and understand why this is. The next step would be to use it proactively, to understand I'm somewhere here in this kind of local minimum, but I'd like to really go up one of the cliffs, but I just don't know where it is.
Yeah, but what I have seen in most of the papers that only numbers are published in a quite tiny range on the P activity scale. And the first measurement in the screening battery is when you are in a lower nanomolar scale, then you run a screen for 100 nanomolar, and if it is lower than 50%, you do not measure the IC50, since that's not needed anymore. And then you will see in the table kind of an activity cliff. You lost the activity. However, the drop was just 3 kcal.
Before we get too detailed, sorry, discussions, I think we have time for maybe one last question. And I would want to put you, Guido, on the spot. Now you get nervous, right? asking you, why is the residence time not more often used, in your opinion, in optimization of molecules in big pharmaceutical companies? So what's your perspective? Why don't they do this stuff?
You precisely gave me now 5 seconds to think about it.
Come on.
So the first can think of is that it's a relatively expensive technology. It's a difficult technology, so it's really asking for specific expertise. I think that's 2 major reasons. There's also still the discussions on what is the added value as compared to just measuring affinities in binding experiments. What I saw as one of the great advantages of looking at target residence time is that it gives a very precise measure of how a compound acts in comparison to, in particular, enzyme activity assays, where you often encounter the limit of detection, in particular if you look at strong interactions. I would rather, from conceptually, target residence time, for all the reasons that there are, but also many good compounds. If you only look at them in traditional assay, you really encounter the limit of detection. But you gave me 5 seconds, and I gave the honest answer. It's complicated and expensive.
Thank you very much.
Thank you. Can I have one short comment?
Please. But no more logarithm.
I will try. As far as I understood, but it's probably a question that if the K on and the K off is different, then the measurement time, so the incubation time, should also vary. We have seen this on a target MCHR1, that if you measure the IC50 after 5 minutes of incubation or 20, the IC50 will change, and I think that's a clear benefit of measuring kinetics. It's a practical, since the IC50 might vary if you measure at different incubation times.
I just want to shortly comment still on the residence time,
Sure.
because no, it's indeed, as you say, it's not often used, and indeed because it's expensive. When as a company, a small biotech, you need to outsource it. But then when I saw it being used, it was mainly when you have your in vitro data, your binding affinity, and you have done all your ADME, and you know that your DMPK profile, it's all nice. But then when you go to your in vivo model, that it's not doing what you expect. And then you go back and you start looking at binding kinetics to try to understand, but why doesn't it translate in what I want to see, to try to understand if the key on or the key off, what could be a better molecule compared to another one. But it doesn't always then lead to the explanation as well, what you would like to see.
Okay. If I can...
Yes,
if I can
that was the reason why I asked you.
Yeah.